Welcome, everyone. So we'll now start our precision oncology panel discussion. And so I just thought I'd just take a moment to set the stage. So the idea of precision medicine is actually to use diagnostic testing to then tailor therapy decisions for individualized patients. And actually, the idea of using this in liquid oncology is not without precedent, as the first idea of precision medicine actually was in chronic myelogenous leukemia, when in 1998, the small molecule drug imatinib was used to target the BCR-ABL translocation for CML therapy. And then, of course, as we learned more about the human genome through the human genome mapping, there was more and more targeted therapies that became available. Now we have therapies that target tyrosine kinase pathways, other signaling pathways, aberrant proteins, immuno-oncology pathway proteins. So we have expanded the possibilities of precision medicine. And today we'll hear about some novel precision medicine drugs. And with that, I'll turn it over to my co-monitor, John.
Thanks, Beth. And excited to be here with this esteemed group of panelists. And while we go through and talk about precision oncology, you'll start to notice that each of these companies and gentlemen have their own approach. Maybe what we'll do is we'll just go down the line and start with Adi and talk a little bit about what it is that you're doing at Affimed, and then we can go to Troy with Kura and Barry at Sapiens.
Thank you. So as said, my name is Adi Höss. I'm the CEO of Affimed. Indeed, we're a German-based company, but since many years listed on US NASDAQ. And what brought us over is the way how we approach oncology, in particular liquid tumors. It comprises mostly bispecific antibodies that we then use to retarget the immune system, in our case specifically the innate immune system. And a way in order to enhance efficacy is that we use the bispecifics now in combination with allogeneic natural killer cells. So that has produced quite some intriguing response rates, and we can do this for a number of targets, and obviously we're going to discuss that further on, how this relates to precision medicine.
Great. I'm Troy Wilson, the president and CEO of Kura Oncology. Beth, John, thank you again for the opportunity to participate in the panel, and thanks to Bank of America for doing this amazing event. At Kura, relevant to this discussion, we are pursuing a class of compounds called menin-MLL inhibitors. These were discovered from a particular subtype of acute leukemia that is very common among infants and children. Children can be born even in utero with essentially full-blown leukemia. And over the last, sort of 2 decades, people have unraveled the biology and then figured out that you can target this protein-protein interaction with small molecules. And we're now at a point where, just in fact last week, we presented data from a phase 1b study where we're showing significant anti-tumor activity in patients with NPM1 mutant AML, which is the largest genetic subset of AML. And that approach has really, we've made tremendous progress in acute leukemias. There are a number of genetic drivers, and we're really delighted to be moving our programs of domenib forward to try to create value for patients. So we'll talk much more about that.
Thank you. I'm Barry Capel. I'm the founder and CEO of Sapiens Therapeutics. And yes, thanks Beth and John and Bank of America and Mass General Brigham, where actually my first real foray into science was at the Brigham in the mid-90s when I got to work as a college student in a lab there. It's great to be here today to talk about Sapiens. Our approach is not really target specific. Of course, it gets there with our different molecules, but what we've done is been able to engineer peptides to stabilize them and deliver them systemically so they can travel around the body and get inside of cells into the cytoplasm or the nucleus, and then bind to a specific target of interest and disrupt protein-protein interactions as well. By doing this, we can go after regulatory pathways, transcription factors, and our first sort of foray into this was targeting something known as CEBP beta with our program ST101, which we started a couple of years ago. We had a large set of data in solid tumors as well as acute myeloid leukemia. But as the smallest company on the panel, for us, we started with the solid tumor approach, but hope to generate data there that will then allow us to move back into the liquid tumors and go after AML at future dates. And subsequent programs, such as our program targeting beta-catenin or our future programs targeting June or FOXP3 and other important both oncogenic and immune drivers of disease, will allow us to go after various liquid tumors with this approach.
Thank you. So I guess the first question I'd like to ask, once that we know a little bit about each of your products and about your companies, is about how you incorporate diagnostics. Because I think that targeting these different proteins and different pathways, we need to be able to identify them in the patient's population. So a little bit, maybe you could discuss about how your approach to the diagnostics is before you develop your drugs.
I go first. So one approach we're targeting is the CD30 antigen known to be expressed in a number of lymphoma. Everybody is immediately targeting Hodgkin lymphoma because Hodgkin lymphoma is encompassing a CD30 diagnosis. But then if you look into further lymphoma, such as T-cell lymphoma or B-cell lymphoma, you suddenly see that the target is expressed at very high levels in some patients, or is not available there at all. And many technologies on top rely on the presence of larger number of receptors in order to be effective. So what we have been doing is we have been in our clinical study in T-cell lymphoma, where you have this broad range of expression, we're using the CD30 diagnostic in a finer way in order to give us a higher resolution from 1%, 5%, 10%, 15% presence, and then testing our drug in these different kind of patients. And what we learned is that our technology is rather agnostic of CD30 expression, and now we can obviously leave it, still we have to diagnose for the CD30 presence, but we don't have to dissect patients anymore. But we only learned it because we were using this, this part of a diagnosis.
So it's a great question. And we think about using diagnostics to help really enrich for clinical activity and to maximize safety and tolerability. Beth, you used the example of imatinib in CML that kind of kicked off the field. There you were targeting a fusion protein and in particular a kinase that's critical to CML. With menin inhibitors, for example, we know certain genotypes are exquisitely sensitive to this mechanism. And what it does, the simple answer is it doesn't kill leukemia blasts, it actually induces them to differentiate, which is kind of a clever trick. It induces cancer to become non-cancer, if you will. The reason we use diagnostics is try to maximize the therapeutic window. So you're looking to find among all patients, those patients who are most exquisitely sensitive, and thereby you give them the most therapeutic efficacy and the best tolerability. And that typically allows you to enrich for activity. It allows you sort of better selection and combinations, but it all starts with how do you find patients. And we use diagnostics in two different ways. We use them prospectively to identify patients, and ultimately that becomes part of the label. And then we also use them retrospectively to understand why, if you have, for example, half of your patients respond, why did this half respond and that half didn't? And that's really, I think, what's allowed this whole field of precision oncology to enjoy such tremendous success over the last couple of decades. It really comes down to the interface between the diagnostic and the therapeutic.
Yeah, I mean, I could sort of take off from where Troy was also suggesting that there are the multiple ways to use them, both retrospectively and prospectively. And what we definitely try and do is start with the retrospective analysis. So when we start our clinical studies, we have a concept that came out of our preclinical work of a genetic sequence or genetic signature that might make cancer cells more sensitive to one of our agents. And then when we enroll our first set of studies, we'll do it without any sort of sorting and no patient enrichment, but allow patients with and without those signatures into the study So then we can validate retrospectively through those patients that we have the right signatures, and then prospectively develop the diagnostic for the subsequent studies to allow that to be on label. You know, with some of our programs, it's more obvious. Our program targeting a beta-catenin or something in the Wnt pathway, we know which proteins or genes to look for. And with some of our other programs where the target itself, even though it's ubiquitously expressed and may only be active in certain tumor settings, that's harder for us to test for, so we look for some of the genes that either drive its expression or genes in which are driven by the expression of that target and try and develop more of a multiplex analysis that can be looked at from a diagnostics perspective, once again, to enrich for the right patients to be enrolled.
You know, one thing that's common between the three of you is AML. And it's interesting, right? I mean, there's been a lot of success in liquid tumors outside of AML. We've seen it in multiple myeloma. Patients have a number of offerings, can go out to 10 years living on some of these compounds. B-cell malignancies also similarly, we're now seeing novel combinations, ibrutinib with BCL2s, but AML's been tough. But you guys are attempting a brave task of going after AML. Question is why? What are some of the lessons learned? And more importantly, how is it that your approach is unique, and what's the key to unlocking this?
I'll let Barry go first this time.
Well, I think compared to the rest of my panelists up here, we're probably the furthest away from approaching AML. You know, it's unique. I was doing some thinking about it with our programs, and there's 400 ongoing phase 1 studies in AML, and there's maybe 20,000 patients per year in the US that unfortunately have AML. So it becomes very difficult from a company like my perspective to think about moving an early program there. But once again, we're going to be mechanistically driven. So we know that the targets that we go after are going to be drivers of AML in the patients we can identify. So when we do go there, it will be very molecularly precisely defined the patient population we want to go after. You know, those that have a specific amplification, whether it's in June or CEBP beta or one of the specific targets.
Maybe Troy?
Yeah. So maybe start with why is AML difficult? And why is it, unlike, John, to your question, lymphoma or multiple myeloma, other areas where we've seen success? AML is genetically very unstable, highly heterogeneous, multiple drivers. You'll see patients who have 6 or 7 mutations at a time. So even if you hit one, another one can sort of take over and drive it. And there hasn't been, unlike lymphoma or myeloma, there isn't an extracellular target that's lent itself to saying, I can target leukemic blasts and spare, you know, spare the normal blood system. And with that being said, you know, the field has evolved, and now we have venetoclax approved, we have chemotherapy, we have FLT3 inhibitors, we have IDH inhibitors. So if you want to make an impact, and this is sort of what Barry was saying, if you want to make an impact in AML, you have to have outsized activity, good tolerability, and the ability to combine with standards of care. It's unlikely that you're going to be a monotherapy and displace the standard of care, at least right away. To the second part of your question, John, why are we doing that? That's actually one of the powers of ziftomenib and targeting the menin pathway. The menin-MLL interaction that we're looking at, it regulates the expression of the key driver genes, FLT3, IDH1 and 2, DNMT3A, plays a role with BCL2, with MCL1, and importantly, it only acts in leukemic cells that have this particular interaction. It doesn't seem to have any effects elsewhere in the body. Once you separate these two proteins, you trick the leukemic blast into growing up, maturing, differentiating, and then after a few weeks it dies off. So it's eminently combinable with all these other approaches. You know, will that transform AML? We hope so. It's early days, but that's the kind of way I think you have to approach AML as a combination therapy.
Yeah, we started out very differently by looking at why the approaches that have worked well for multiple myeloma or B-cell lymphoma are not working so well in AML. So it's the antibody-drug conjugates that had failed mostly because of toxicities, myelotoxicities, myelosuppression. At the same time, the T-cell approaches have not made the same impact either from the CAR-T or from the bispecifics that were seen in the other indications. So as such, we were just like analyzing the data that are in front of you that has been emerging, and a gap has opened up. And this gap to us was that there is another technology needed, and usually people have followed in with antibodies, but we have learned that antibodies cannot effectively activate the innate immune system, and that's how we brought the bits and pieces together as a hypothesis. In parallel, a few people were starting to investigate natural killer cells as a single agent, non-modified cells, and showing that the blasts respond pretty well basically can be destroyed pretty well by natural killer cells, which is a little different to what has been seen in multiple myeloma or in B-cell lymphoma. So suddenly you were realizing that there is an opportunity for innate-based approaches, and what we identified as the missing link is to develop a bispecific that can effectively engage these cells, meaning that we have unprecedented affinities to bind to natural killer cells, forms a very stable complex, and thereby you make the cell far more better attacking the tumor cell. Now, the one question that still was remaining, is this a safe therapy? Also, would it also cause the same side effects on targets that have been used already for a long time, in particular CD123? So why did we end up in CD123? It's not only present on the blasts, but it's also present on the leukemic stem cells. So it goes to the origin, where many relapses may be related to. But CD123 was impacted by a lot of toxicity, and it was described that it is maybe residing within the target itself. We, when looked at that, thought, no, it's modality-driven. It's more caused by the T-cell or by the ADC approaches, but it may not be present on the same end when you use NK cell-based approaches. We now see early data emerging, and indeed the toxicities that are being detected are very different at the moment, almost absent, I should say. So it's only infusion-related toxicities that you see with NK cell-based approaches, but none of the other toxicities are there. And we see the first results emerging that you can generate anti-tumor responses, as such CRs. So the companies pursuing this with NK cell-based approaches are Sanofi on the one side, that just recently had its first data presented at ASCO, and we at Affimed, who are still a little earlier in our dose escalation, but we see a very similar, well-manageable safety profile emerging in the dose escalation. So this is how we got to this field, filling a gap, as I started out, and hopefully that's what we're all sitting up here, is to have some solutions for patients that have an enormously high need.
So, Adi, you brought up some very interesting points, and of course, as someone who takes care of patients, we often see that our patients stop responding to some of these medications, and so I think that I want to hear from all three of you a little bit about how you approach relapse and what the next steps are for your products in terms of dealing with the patients who no longer respond at some point.
Yeah. So I can start out with that again, going back to lymphoma, where we now have the most data. And so this is where we first tried a bispecific innate cell engager in combination with an allogeneic NK cell. And when we treated the patient with a single cycle, so once time giving the cell and then following up once weekly just with a bispecific in order to re-engage, we were seeing that still in a small patient number at the beginning, every patient responded, and about 1/3 of the patient had a complete response. Then we gave a second cycle, and the complete response rate jumped up to above 70%. So unprecedented data, all generated, I should say, at MD Anderson with the help of Iago Nieto and their cell, their NK cell. But we were very surprised that we could generate these deep responses. Now, what we learned in order to prolong any of these responses is that we have to give the modality over a much longer period. In contrast to T cells, where you try to engraft a T cell, you're not achieving the same with an allogeneic NK cell. It's more considered to be an antibody-like therapy. And the key question here is, in order to give an NK cell, you need the lymphodepletion. So what we did is we went with a very mild dose for lymphodepletion that's not affecting and causing any, let's say, medical safety issues for the patient. So we are far lower in our lymphodepletion than what people are used to use for T cells. And this enabled us to repeat these lymphodepletions now 4 times. So you can give up to 4 cycles, and indeed, we could have gone up to 6 cycles. So that means that we have a much more prolonged treatment period. We're now extending a cycle to 12 weeks, doing it 3 times, but you can extend it to 4 times, and it's very safe. So this is, as long as you treat a patient, it seems to be stable with an NK cell, and that's how you prolong the response rates. That's the way how we have done in in lymphoma, and that's what we will apply to leukemia as well.
So it's a good question, Beth. You know, the title of this panel is the innovation gap. One of the things that's frustrating, I think, as a drug developer is, and it's understandable, it's the time required to do the experiment. If you think about the cycle time in this industry to generate data, analyze that data, and then do the next experiment, it takes years. Versus certain other industries where, essentially anything on the internet, you can get data almost on the fly, real time. We start in the relapsed refractory patients because, number one, it's an area of extremely high unmet need, and by definition, you start with patients who have exhausted all available therapies. Safety is paramount, right? You would tell us that, and that's absolutely the right focus. That puts the bar so high that if you see a signal there, you have reasonable, and you have good tolerability and encouraging activity, it's reasonable to believe you can move it to earlier lines of therapy and the signal to noise will increase. For example, we're treating patients who have failed a median of 3 prior lines of therapy. Their life expectancy, this is our NPM1 mutant AML, their life expectancy is about 2.5 months in the relapsed refractory setting. The median duration of response at this point for our phase 1b trial is 8.2 months. So that's pretty clear signal to noise, right? What you'd like to do is, what we've seen many times is when we lose patients, it's not because of the leukemia, it's because they have graft versus host disease, or they have other complications. They've exhausted so many therapies that these patients are just so frail. But that is what we have to do. Interestingly, the FDA, under its Project Frontrunner initiative, is trying to encourage companies now to go earlier, take bigger risks, think about how we can design trials that are seamless. And I think that will help to close the innovation gap. As an industry, we're very good at generating molecules that have therapeutic potential. We're now trying to do 3 things simultaneously. We're going to complete a registrational study in relapsed refractory NPM1 mutant AML. We are starting frontline studies, and we will shortly start maintenance studies. And if you want to make a difference, you have to tackle, you want to establish yourself across the continuum of care. But that's a big financial risk, and it means for a small company like us, you have a lot of plates spinning at the same time. But a combination of good development, working with the physicians, with the regulators, with pharma, that's how we're going to close that gap.
I think with our therapeutic approach, I'd like to say we were far enough along in any of our clinical development programs to see the relapse and refractory curve. But like Troy said, we start with these very late stage patients. But I think our therapeutic approach being larger than small molecules allows us to avoid the potential of some near-term mutants to escape our therapeutic approach. But I also think as we move forward in development, having the secondary mechanism, so our very specific approach, marrying it with maybe a broader systemic approach, even if you can lower the doses of other drugs to put in combination, will maybe give better coverage of our agents to allow, first of all, synergistic activities based on mechanism, but also to avoid escape based on any single one of those avoidance pathways that a type of cancer can come up with. So I think we think about it just as multi-modalities at once, maybe one precise, maybe one a little less precise, that can really add to one another in the clinical setting.
I guess alongside that, I guess the corollary is off-target effects. And one of the key concerns is with new modalities, new approaches, new targets, off-target effects. And what are your thoughts with your approaches now, and what are you guys seeing in your respective programs? Who wants to go first here?
I'll take that.
Yeah.
So when you start a clinical program, you have a 95% chance that program is going to fail at some point. There is no other industry where you can tolerate a 95% failure rate. Like, think about aircrafts. If 95 out of 100 fell out of the sky, you wouldn't have a viable business. It is when that 5% succeeds, and even with all the innovation, that number hasn't changed tremendously because we keep raising the bar, right? That's why that number stays about where it does. And John, to your point, it's relatively rare that programs fail for lack of efficacy. They typically fail because they're due to unexpected toxicities or toxicities that just are insurmountable. And the reality is we do all the best preclinical work we can, but humans are so heterogeneous, and human physiology is still so complicated. Even with AI and other approaches, when you dose those first few patients, you hold your breath. You really do, because nothing ruins a good plan like data. What we've done is you de-risk it, obviously, preclinically, and then you learn as you go. And it is having tremendously talented physician scientists who are experts at phase 1 development, who understand, okay, how do I manage dose? How do I manage schedule? How do I think about this? It's rare that you can take a drug. Almost anything you put in your body is going to have some measure of toxicity. You just tolerate it, right? Think about caffeine as an example. You get a headache, that could be a dose-limiting toxicity if you were in a trial. But that is what it is. Your physician scientists help you engineer it. With our menin inhibitor, that's actually been, I think, the biggest breakthrough we've seen is the drug class, our drug in particular, is extremely well tolerated. But it's taken us, we've dosed now more than 80 patients to really get a good understanding. And it just, in full disclosure, we were on a partial clinical hold last year because we had a grade 5 event. We had a fatality on mechanism. That could have very well killed the program. It was Dr. Erba at Duke University. If I had AML, I would go to Dr. Erba. He figured it out and he rescued it. And at this point, I think it's off to the races. But it was his talent that got it done.
From our perspective, we think about this from target nomination, from concept nomination, when we think about what we even want to put into our discovery program, our discovery platform. One of our key criteria for a target is that we believe either there would be no on-target off-tissue effects of targeting this in a normal adult, or that we can bifurcate that activity. So if we think about our two lead programs, ST101, which targets a transcription, an embryonic stage transcription factor known as CEBP beta, even though it's expressed in all of us, it really has no activity in any of us. So targeting it in a non-cancerous cell will lead it to proteasomal degradation, but it'll have no downstream effects because it's not driving genes transactivation in any of our cells. That program's now been in about 100 subjects, and we also design our molecules to be non-degraded themselves. So they're very well tolerated. We've had nothing at all above a grade 2 adverse event. They're non-immunogenic. And they've really proven that concept of really no on-target off-tissue effects. Whereas a second program of ours goes after a very important physiologic molecule in a normal, healthy adult, and that's beta-catenin. And before we even considered that as a target, we challenged our scientists to come up with a target on that protein itself, that if we can bifurcate the pathological versus the physiologic activity by disrupting particular interactions while leaving other ones intact. And I think that's something that's amenable to a technology like ours, a peptide, which can bind to part of a molecule Whereas if we were trying to take out the entire pathway holistically, we know a lot of Wnt inhibitors in the past have led to rampant off-target, on-target, off-tissue effects, such as affecting the gut axis or the bone formation. And all those programs end up being killed in clinical development for those. I wish I had 100 patients of data to talk to there. I can say we've dosed 2 patients so far. So far, so good, but it's only been a couple weeks. We're excited to continue to report out on that program, but preclinically, non-clinically, we have shown that we can indeed bifurcate these effects. So we think about it from day one scientifically, and hopefully it carries through into the clinic.
I have to be very scientific because I like your comment. An industry that fails 95% and still surviving. And I was thinking, is there anything else? And we just talked a little bit about Italy before, and I'm just telling you, the world is failing on preparing good pasta for the reason they're always overcooking it. So 95% is probably not knowing what pasta is about. When you go to Italy, it's very different. And the reason is not it tastes different, it's cooked different, believe me. 95% of the world believes different. It's just one example. But coming back to, yeah, sorry for this detour, but I had to make this here. But our point was an interesting approach. Again, coming out of immunology, you see that the cells that are comprising our immune system are very different ones. And we have the NK cell there that is not target specific, so it's controlled. And it still recognizes the good cell and the bad cell. So there is something intrinsic that you can use to your advantage. And we were then starting off with targeting NK cell through our bispecifics. As I said in the beginning, the CD30 is truly tumor specific. CD123 mostly is, but then we jumped far away from this tumor specificity and took a promiscuous target, EGF receptor. And here the question that came up, okay, let's prove if this, what we know from NK cells, also can be made true in oncology, that this targeting would only activate itself in the context of a tumor cell, where the tumor sits, but not attack the healthy body. Obviously, you can test that in your animals, in particular in tox studies in cynomolgus monkey, because many of the targeting agents in EGFR showed its toxicity in the monkey already before when they went into humans. So this could somehow be replicated. So what we did is nothing else than early on tested such a bispecific innate cell engager in a cynomolgus monkey and didn't see any of the toxicities. So none of the healthy tissue has been attacked. So this was the monkey. So we went into the human body, did our dose escalation, and it's been confirmed. We have no MTD, still see a specific activation of the innate immune system in the first place. The efficacy is modest, but that doesn't relate so much to the power of the innate cell engager. It's mostly because the patient itself does not have sufficient natural killer cell for different reasons. We discussed that we are treating mostly end-stage patients that have undergone a number of therapies and their immune system is impacted by these therapies. So essentially, we have to basically fill the body back with allogeneic NK cells, and that's what we're trying next in order to test these agents in that combination. But what we have learned is you can drive, or basically you're using the gift of nature that NK cells are largely activated, or mostly exclusively activated in the context of your tumor, and thereby you avoid the respective toxicities. That's how we have explored it, and you said it before, it takes you years until you finally are there, from starting of the design until testing it in your tox models and finally testing it in the human body. But this is what our approach has been about.
Wonderful. So I'd like to take us back to this idea of innovation and gaps that are hindering our innovation. So not only do I spend time in clinic, but I'm a physician scientist, so I spend a lot of time, and it's called the luxury of my day, is surrounded by amazing scientists who are developing new treatments and discovering new things. And I guess I'd like to, this is a little bit of an off question, but I'd like to understand a little bit about how we can close the gap between the academic side and the technology side. So how could we bring some of these things faster that are happening in the lab into our industry side of things so that we can make things better for patients faster.
I mean, for us, when we started out, we wouldn't exist as a company if there wouldn't have been academic research in the first place. Okay? That's very clear, and where we are enormously profiting off is that, in particular, academic centers have started to investigate the effect of NK cells as such, and moved it forward and forward in order to become a product. However, and that's where the gap has been emerging also for us, is an academic cell can help you to produce clinical data, can help you to establish your proof of concept, but it is not a product that is ever reaching the market. So that's when manufacturing, all your CMC comes in, and there is a gap. And this gap needed to be filled, and that can take a long time for cellular therapies. In particular, when it comes to allogeneic cells, you need quantities. That's important. You need not only the quality, but also it's just like a technology that needs to move from small scale, where you produce cells in a liter, and then you move into 50 liter. So from the beginning until you have that, you may have to invest another 3, 4, 5 years. And that's really been something that's been challenging for many companies, and only a few are there at the moment that have filled this gap.
And I've been fortunate to start several companies initially working with universities and technology. But I think that it's very important when you think about how to get technology from universities successfully is that it is mature enough. I think privatizing late-stage academic science can be dangerous to the company. You know, when you get some big investments in things that aren't ready yet, you know, you get those big failures. And those, I think, really hurt funding opportunities going forward. So I think that's one of the big challenges is letting it mature enough, and there's that fine line, you know. And then I think, even though I mentioned I've started several companies through academic collaborations, then there's always that tension at the end of the collaboration, because the academic scientist has his own dreams and pursuits and continues on in that path, eventually creating his own competitor. So it's a great way to jumpstart industry. It's a great way to do things, and hopefully you have a close enough tie with the professor, so that they continue to work with you and really push that particular program forward. But it is, obviously, it's a driver of such great science and behind so many companies.
So one of the critical things you need in that gap between academia and sort of early industry is risk capital. And particularly in the context of this panel, I want to acknowledge the Leukemia and Lymphoma Society. They have had a profound impact on the treatment of liquid tumors. The last number I saw, you know, of the 20 therapies approved, they've played a role in funding like 17 of them. Or I don't know if I have the numbers quite right, but they played a specific role in the development of menin inhibitors. And I was working in 3 companies ago doing the KRAS mutant work as part of Wellspring Biosciences, and I got a call from Lee Greenberger, the chief scientific officer at LLS. And Lee said, Troy, there's a couple of scientists at the University of Michigan. We've been funding them for 6 years, we LLS, on unraveling these mixed lineage leukemias. They have an interesting molecule. They think they have a drug. They need some drug development expertise. Would you be interested in taking a look? And, you know, Lee's a friend, and I said, absolutely. Went up there. They didn't quite have a drug. You know, we took it back into the garage. We took it apart. But ultimately, it was LLS funding their effort for 6 years. And then we've put, you know, I don't know, $100, $200 million into that program. They were the ones who paid for the risk capital. And it's their charitable donors, it's their expertise, it's their networks, it's knowing how to pick projects. If you can do that on a problem by problem basis, that really helps close the gap.
Wonderful. Thank you.
Well, I think we're basically out of time, but I want to thank all 3 of you for helping us and better understand the innovation gap and what it is that you each are doing, and wish you continued success.